We proceed with the second presentation, which I believe is a joint presentation by Peter Kharchenko and Ed Lane about cell type annotation, a topic which is of interest to everybody in the audience.
All right, terrific. So first of all, I'd like to thank the organizers for the opportunity to speak here today. Peter and I are going to do a little tag team presentation. We were asked to split this 12-minute segment on the topic of annotating cells. I have to give Peter credit for the title here, On Your Own with Friends or with Everyone Else. And so I think we're going to have two slightly different takes on this. I'm going to talk first about brain. I think in the spirit of sort of creating this Leinion classification, we're kind of in a remarkable time now where the most complex organ is also perhaps going to be the first to get a really comprehensive coverage. And this is in large part due to the generous funding of the NIH Brain Initiative and the Cell Census Network. We're very quickly now, we're starting to get a remarkably detailed understanding of the different transcriptomic types that make up different parts of the brain. This is moving so fast, it's moving from a publication on 2 regions of the brain 1 year. A year later, we'll have pretty much the entire brain. So this really gets us down to the nitty-gritty of trying to understand this data and trying to actually make a reference out of it. One other point I wanted to make is that a lot of the early progress is actually in the mouse, which is then being followed by the human. But it's turning out that the homology is so strong between the two, you can really align these. And so there's a great value in having both of these and in co-developing these things together. So as we get this data, it's quite remarkable to have a look at the diversity of types, which is far more than most organs. There are likely to be thousands of types. And everyone is seeing these things and calling them something different. So just in the neocortex, there have now been a suite of papers that profile diversity in different parts of the cortex of mouse and human, and they all come up with different numbers, and they all come up with different nomenclature systems. And it's a bit of a mess. Really, the promise of this is that we can come to a reference and a standard and convention of what we call these types of cells, somewhat like the genome. Like we can have RefSeq, we should have some version of cell classifications that serve the same purpose so that people map their data to the reference rather than creating their own de novo classification. And of course, this needs to be iterative so that you take new evidence into account. So, I think this really creates some remarkable opportunities here. In a field which has been dominated by somewhat qualitative classifications, we have the promise of now a quantitative way of defining the vast majority of cell types and states across the brain and across every other organ. By sticking in the transcriptomic space, we can compare across any organ. The brain is one organ among many. As of course, I've come to really appreciate through this community here, that we'd like to have something which doesn't just work for brain, it works for all the different organs. You can quantitatively align across species so that you can call things the same way, and even across developmental trajectories as well. So of course there are many challenges. This is a small subset listed over here of how to quantitatively define types, how to accumulate evidence. Peter's going to talk about this, that they're meaningful. How to develop these cross-species consensus classifications and nomenclatures, just like gene symbols, so we call things the same thing across species. And also how to get out of sort of an abstraction away from calling things from the primary modality from which they were generated. An example of this is marker genes are very often not the same across species. So you can't call things by marker genes if you want to call things, talk about the same things. Eventually, of course, we need to get to a full formal ontology system. So as a start for this, as we're dealing with these data, we've been trying to develop a flexible nomenclature system that can work with these different types of data. And part of this is trying to sort of learn from lessons of the genome that perhaps can be applied at the level of cell types. So for example, the idea of an iteratively generated reference classification, the idea of unique sequence IDs that could perhaps be turned into cell type accession IDs, many aliases. We have lots of names for things. We also want to bring in sort of common usage names into this as well that are defined on different modalities. And so we've developed the beginnings of a system. I should say this is not by any means perfect. This is meant to be a draft that can be improved over time with the community, I hope. where we have, of course, taxonomies. There can be many taxonomies. There's the classification based on particular data sets. We introduce the idea of a cell set. At one level, this can be a cluster, but it could be larger aggregates of cells in subset kind of relationships. At the sort of finest level, maybe a provisional cell type would be a cluster that is a molecularly defined cluster that hopefully turns into a meaningful functional cluster as well. And then we're trying to apply some of these ideas of accession IDs to cell sets and aliases of different ways of naming these as well. And labels as sort of a more abstract version. So to give you an example, here's applied to one of our mouse cortical data sets. This particular type of data set is actually quite well modeled in a hierarchical fashion. And so we can define cell sets that are either the finest clusters, the putative cell types, as well as higher order nodes. And those nodes can then be named and they can be aligned with common knowledge for things. So for example, glutamatergic neurons or GABAergic neurons, things like this. We can name all these things and define all these different nodes. So this seems to work quite well. The other thing is that as we have these very large data sets, we're now starting to think of extend this beyond data sets to a reference classification. And this would initially probably start with these very large monolithic data sets being generated in the consortium, but over time you want to integrate other information. And so these will become increasingly refined as there's increasing evidence, or more cells give you more types and things like this that you can continue to refine and iterate on this so that it represents the current state of knowledge. And so in the interest of time, I'm going to sort of end here by saying that we're trying to do this in a very open way. We've had a workshop. We're test driving this whole system on a big data release that we're actually having next week. The whole rationale behind this and even code for applying this to your own data is available on GitHub, and we have a community forum. We would love your feedback on this to see whether this would be something that could be used across more sets than this, more organs than this. And finally, I'd just like to thank a few of the people in particular. Jeremy Miller, and not seen here, are Amy Bernard and Mike Harlitz have really been instrumental in developing this.
Thank you. So it's quite a team here, and we thought it's actually important to think as to how these annotations may be accumulated and perhaps reconciled at different levels. So if, for instance, now in Brain Atlas, there's this new exciting piece of the brain, Ed might jump in himself and actually annotate much of it. However, Hangui might have, additional corrections and so forth. And of course, Basilica will certainly point out some mistakes. But they're all part of the same team, and as that just showed, they're working to come up with a process, with a nomenclature and sort of a structure that would allow them release sort of a uniform annotation to everybody. And for most of us mortals, this will be enough, and we'll probably follow this. But there will be people who will disagree or want to see more detail, for instance, in oligodendrocytes or perhaps in microglia and so forth. And if the data sets are public, as we hope most of them will be, this information, this alternative opinions or more detailed classifications will be available there. Now, some of these alternative opinions will be more useful than others. Someone might jump in and actually annotate, let's say, cell cycle phases in this data, even though it doesn't relate to sort of cell type annotation. So overall, it would be nice if there was a system to kind of resolve some of these differences, view these alternative opinions, and allow to sort of explore this data and annotate it jointly. Of course, it's important to assess the value of these different contributions so that useful and consensus annotations kind of bubble up to the top. So some kind of a ranking perhaps and eventually consensus convergence and some consensus nomenclature would be useful. So in thinking about all this, we propose to build a system called Central Annotation Platform. Essentially, it's a cloud database that stores annotations at its heart, but to make it useful and compelling for people to use, it would bring in a rich set of user interfaces to actually view and deposit annotations. It would also serve as a directory for sort of automated annotation tools that different groups may develop. Important that it doesn't pretend to store the data itself, right? It stores only annotations, and the data itself, let's say, sits in the DCP and so forth. So just kind of quickly, there are increasing number of different graphical user interfaces that make it very convenient to explore this data, to look through markers and perhaps identify subpopulations that are of interest. and experts, biology experts in the tissue can use this. So what we're proposing is to essentially build this up into sort of set of module interfaces that can be integrated with all major frameworks for single-cell analysis and perhaps portals to first of all allow viewing for a given data set, allow viewing of annotations. So you would have basically a list of annotations that have been deposited by different users or groups perhaps, and you can selectively view them and try to make sense of it this way. They're ranked, so you could prioritize them or filter them and so forth. By annotations, we basically mean simply a term and some kind of association of that term with a set of cells that are available in the data set, maybe probabilistic, maybe some evidence history. But the main thing is storing the label associations with the cells. Now, if such data accumulates, a lot of very interesting analytics can be done on this. We can infer automatically term relationships, which terms are basically synonymous, which ones are, fall into some kind of hierarchy, and perhaps rank them by usage and so forth. Furthermore, you could envision this type of cell cards, kind of like gene cards page, which would essentially just allow you to explore, let's say, other data sets where these cell types appear and so forth. The key aspect of all this is actually entering annotation. So here, if, let's say, a biology tissue expert is looking at the data and has additional ideas, and they start to annotate it, of course, we can't tell them how to annotate it, but we want to encourage them to use common vocabulary, and we want to give them hints if we can. And so the main aspect here is that behind the scenes, once this cluster is selected, we can already analyze some of the molecular signatures there and perhaps come up with some molecular-based predictors. Sorry, I'm losing a pointer here, that the machine can guess. Now, we might be wrong, it's just a suggestion. Obviously, as a person is typing in text, we could look up sort of, not just out of completion, but actually synonymous terms to what a person is entering that are more commonly used, again, to encourage them to converge to sort of a common vocabulary. Finally, we can come up with some suggestions for, let's say, potential child terms, right, to kind of encourage people to annotate in a more detailed way, not just, T lymphocytes, but a more specific subtype. And finally, as I mentioned, we would like to have the system work as a directory for automated annotation services, which many groups are developing currently. Of course, all this lies on the assumption that we'll get informative contribution, there'll be some community cohesion, but HCA community is excellent in this regard. There are quite a few challenges, I think, and opportunities in terms of analytics here, in terms of text processing. The huge advantage we have here over all kinds of crowdsourcing efforts is that we have the actual molecular data, which should allow us to resolve a lot of issues and evaluate, let's say, the quality of annotations or consistency of annotations. But of course, building these module visualization tools and integrating in different frameworks, that will take quite a bit of work. But we hope that with this effort, basically, we'll be able to create this kind of open annotation cloud infrastructure, user-friendly interfaces, which would eventually provide resource for both machine learning and for biologists exploring this type of data. And of course, I wanted to thank all the people involved in this, Aviv, Fabian, John, as well as Anna Hupalovska, who helped us to actually visualize a lot of the mock-ups that I've been showing. Thank you very much.
Yeah, thank you very much for giving two very different facets on one and the same topic. Questions to add, and Peter. So while he's running up there, Peter, we all talk about annotation as a static thing. So what if, the cell type changes, over time under different conditions, but it still should be considered the same cell types only under different circumstances or different modes. What about taking this into consideration?
Yeah. So I kind of alluded to this briefly that you don't have to limit yourself to types. You could start talking about states and conditions and so on. And again, if we're careful about allowing this ranking and prioritization, people can do both without conflicts arising. Eventually, I hope we'll arrive at at the computational methods that will be able to distinguish these axes, of
absolutely.
state variation and so forth. But initially, we need this information from experts in order to even start learning this. So this is essentially just a way of centrally soaking up this type of information.
So there was the first question up there.
Peter, I have a question about the hierarchy you mentioned, and Ed also. So sometimes when you're doing it de novo from the data, right, you can see some clusters, but if you look at it this way, this cluster is part of this hierarchy. If you look at it that way, this cluster is part of another hierarchy.
Yeah.
And so is there really a ground truth hierarchy for these cell clusters that we can define and all agree on?
Right. Right. I think certainly there are cases where we know the differentiation path and there's a clear hierarchy. There are other cases which clearly cannot describe in a hierarchical manner. So we'll have both. The little view of snapshot I showed, I mean, essentially those would be inferred relationships from the data, from what people are contributing. And if it looks very hierarchical, we can show this as a sort of as a relationship. If it doesn't, then it will be a more messy graph essentially.
Okay.
Fabian?
Question to the both of you. Do you think you can annotate a single cell, a region, or maybe all of its entity? Essentially, if I add something to the very right of my t-SNE, is my annotation on the very left influenced or not?
You want to take it first?
If you have one cell, would you be able to annotate?
So in the brain, for example, right? Do you annotate single cells or like whole regions?
So I certainly hope so, because the way to connect phenotypes here is to measure individual cells with multiple modalities. So, for example, one strategy that we're taking for that is a technique called patch-seq, where you actually record physiologically from a cell, you fill it with a dye, and then you extract the contents of the cell and you sequence it. And the assumption is that we'll be able to map that back onto the reference classification generated from cells that we didn't measure other things from. And it's absolutely working. It's a little bit noisy, but it's definitely working. I think the shallower the method gets, the harder that is to do accurately. So, that's one place where we haven't gone to 10x, for example, because we can use SmartSeq and get a much more rich reading and a better mapping. But yeah.
And I think from CAD's standpoint, we're not trying to make any assumptions like this. The only thing we know is that a lot of times, most of the time people annotate clusters, so we'll keep track of, what was a cluster. You can infer, more detailed profiles from that. But if someone can actually annotate a single cell, sure.
Could I just make one comment? Because I think these things really go together. The diversity of types in the brain, for example, is so huge, and we actually don't know what most of them are. We really don't. And so experts can't come and annotate it yet. We actually need people to be generating data to put some annotation on there and then contributing it to that. And that's going to be a giant community effort, I think, to do that.
Yeah. Okay, thank you very much again, Peter and Ed, for fascinating talk and discussion.